I would like to tell you about some of our recent work trying to address the kinetics of splicing, how fast splicing happens in the nucleus. That's the main question that we have been trying to answer. And why are we fascinated about RNA splicing? I would like just to highlight that RNA splicing is intimately connected with the evolution of more complex organisms. And it's still quite intriguing why introns have expanded by thousands of nucleotides during vertebrate evolution, because we all know that introns are there to be removed, to be excised by splicing. So why care to let introns become larger and larger? Because in fact, humans, as we look at the vertebrate genomes, humans have the longest introns among all the sequenced vertebrate genomes. Why? So why do we have so long introns, and how come this extremely long sequence of RNA is correctly recognized and excised from the pre-messenger RNA? And splicing is also intimately connected with disease. More and more, you name whatever disease, when people look at splicing in any type of disease, it's more and more common to find abnormalities in splicing patterns. There is also a very large proportion of mutations that either cause disease or modify disease that are located in introns or act by affecting splicing regulation. And now we already have in the clinic some RNA therapeutics that target the splicing machinery and the splicing reaction. So the connections between splicing therapeutics and the clinic are growing and growing. So most of what we know about the mechanism of the splicing reaction was obtained many years ago using in vitro splicing systems that basically are done in a test tube. And this test tube contains nuclear extract that is prepared from isolated nuclei. And then an in vitro transcribed RNA is added to these nuclear extracts. And the result is that the initially added pre-messenger RNA becomes spliced into a messenger RNA. And an obvious conclusion from these experiments is that splicing is independent from transcription, because the splicing reaction has occurred on a pre-transcribed pre-messenger RNA, so a pre-transcribed RNA. But what also become evident is that in vitro, it takes quite a while to splice. So when people do time course analyses of this in vitro splicing reaction, it takes between 20-45 minutes to accumulate spliced products in vitro. So as soon as technologies and approaches to look at splicing in vivo started to be used, the scenario changed quite dramatically. Namely, because it became obvious, and this was one of the first and very striking, very visual evidence. It became obvious that splicing occurs as transcription is taking place. So these are electron micrographs of pieces of DNA, so genes that are being transcribed by Pol II. And this has obviously been, the nuclei were isolated and were swollen so that all the chromatin becomes decondensed. But what was obvious to Ann Bayer and Osheim back in the 80s was that they could see in the nascent RNA molecules, they could see these lariats that they interpret correspond to introns in the process of being excised. And this becomes very obvious when they measure the linear length of these RNA molecules. You can see that this RNA molecule is long, probably because it still contains an intron. And when the polymerase transcribes a little bit further, now the RNA molecule becomes shorter, probably because the intron has already been excised. And by measuring the distance that the polymerase moved from this point to this, and taking into account the speed of the polymerase, what these authors concluded is that splicing may occur within 2 to 3 minutes after transcription, which is in striking contrast with what happens in vitro. So in vivo, the splicing reaction is much more efficient, and probably this is related to the fact that splicing takes place as the polymerase is transcribing the gene. But how can we move from these beautiful electron microscopy images to a more precise and genome-wide view of the dynamics of splicing relative to transcription? In order to address this question, we started now some years ago a collaboration with the lab of Nick Proudfoot at the University of Oxford, and a postdoc in Nick's lab called Taka Nojima was adapting a technique called native elongation transcript sequencing, for short, Netsig. Taka was adapting Netsig to mammalian cells. Why there was a need to adapt? Because the technique was originally discovered and developed in the Weizmann lab by Sterling Churchman, and they used yeasts, and they used polymerases that were tagged, that were epitope tagged, and this allowed for them to perform immunoprecipitation of the polymerase. So basically, what this technique is all about is to purify by immunoprecipitation the polymerase, so Pol II, as it is transcribing pre-messenger RNA. So what is the basis of the technique? You start with a preparation of chromatin. And now the big challenge is to solubilize the polymerase with its attached nascent RNA from the bulk of the chromatin. This is relatively easy to do in yeast, but it is much more complex to do in mammalian cells. And that's where Taka introduced an innovation which consisted of digesting the chromatin prep with MNAs. So MNAs will cut the DNA, will also cut the RNA, but will allow to get soluble complexes containing a bit of DNA, the polymerase, and the nascent RNA still associated with the polymerase through the last nucleotide that has been incorporated as the polymerase was elongated. So once you have this solubilized, you now can use antibodies against the polymerase to pull down the complex and either look at the proteins that come together with the polymerase, or just destroy all the proteins and just isolate the nascent RNA and do an RNA-seq analysis of these nascent RNAs. So our idea was to look when splicing occurred as the nascent transcript was still attached to the polymerase. But our initial attempts, although the technique worked and we were able to identify several features of co-transcriptional splicing, we were not able to detect the splicing reaction taking place in association with the polymerase. As I depict in this slide, we are able to isolate the spliceosome associated with the polymerase. We were able to identify the snRNA components as well as protein components of the spliceosome. We could detect splicing intermediates associated with the pol2, but the ligated products were hardly detected in our initial experiments. But as we were doing these experiments, a very nice paper was published from Carla Neugebauer's lab, and they were addressing a similar question, but using yeast cells. And what they were able to detect using yeast cells was that they already detect splicing transcripts associated with polymerases that were transcribing just after the splice site. So in order to recognize an intron, you need first to transcribe both splice sites, the 5' splice site and the 3' splice site. And then as the polymerase was elongating along the downstream exon, they could already detect spliced, so excised introns within a few nucleotides after the splice junction. So why Carla and colleagues were detecting these fast splicing in yeast, and we were not detecting fast splicing with our Netsig? Well, one possibility is that most yeast introns are very short. In contrast with human introns, I mentioned to you in the beginning that human introns tend to be very long. And so maybe the short yeast introns can be spliced faster than the human introns. In thinking about this possibility, we decided to look at an intermediate organism, and that's drosophila. These are flies. So, and we focused on performing Netsig in drosophila embryos. Why? Because in the very early stages of drosophila embryo development, the genes that are expressed, they tend to be mostly like yeast genes in the sense that these genes are short and they have very short introns. Whereas at late stages of embryo development, so a few hours later on in development, now the genes that are expressed are more like the human genes with much longer introns. Drosophila has another advantage because it has a more compact genome compared to human, and therefore we have a higher Netsig read coverage. So we have more reads that we could focus our analysis on detecting more spliced reads. And I would like to introduce you to the people that have been involved in this work. So Pedro Prudencio did all the experiments. And Pedro was trained in Drosophila genetics by a colleague, Rui Martinho, who is a Drosophila geneticist. And Rosina is a computational biologist that join our lab as a postdoc. So Pedro very patiently handpicked drosophila embryos, separating early stages from late stages. Then he isolated the chromatin, did the MNase digestion, and then did the pull down and isolated RNA and prepared libraries for Illumina sequencing. And we were very happy to detect now spliced reads associated with many genes in Drosophila using the Netsig approach. So this is just an example. This is a gene. You see here a blow up. You see this is a relatively short intron. These are all the reads associated, and you can see, so each of these line corresponds to a read associated with this particular position of the polymerase. I recall that Netsig gives you single nucleotide resolution, so we can tell exactly nucleotide by nucleotide how many RNA reads end in that position, because we are doing just go back. We are mapping the OH end of the nascent RNA, and the OH end is the last nucleotide that has been incorporated by the polymerase. So by mapping the 3' OH, we know exactly where is the position of the polymerase that was transcribing that particular RNA. And so you see, when we look at total reads, you have reads being produced as the polymerase goes, elongates along this exon, along the intron, and then along the next exon. But when we just focus on reads that have already been spliced, so that this exon has already been spliced to the previous, you can see that we start to detect these spliced reads after the polymerase is transcribing the downstream exon. In the beginning, there is very few splicing, and then as the polymerase moves along, the spliced reads become more abundant. So this was fine, and this was basically recapitulating and reproducing what Carla had seen in yeast. But what about longer introns? And here I would like to recall you that in the splicing field, there is this model of intron definition and exon definition that posits that for short introns, when introns are short, it is sufficient for the spliceosome to recognize the two splice sites. And this is sufficient for the spliceosome to assemble and remove these and excise the intron. So intron definition can happen as the polymerase is transcribing the downstream exon. In contrast, when introns become longer, exons and the proteins that associate with exons collaborate with the spliceosome in order to assist the spliceosome to know where the spliced junctions are, given that the intron is very long. So the exon definition model assumes that exons have to be completely transcribed before the spliceosome can remove the intron. And in this case, we would expect that the polymerase is already transcribing past this downstream exon when the splicing is detected. So this was the expectation, but this is not what we observed. So I show you now an example of a gene that is expressed in late embryos, and now this looks much more like a human gene. short exon flanked by 2 very long introns. And when we look at Netsig reads, we start to see spliced reads associated with the polymerase transcribing the exon immediately after the intron has been totally transcribed. So we did a careful analysis of how much splicing, fast splicing, is detecting for introns of different sizes. And you can see that we go from short introns, 40, 50 base pairs, to very long introns with more than 10,000, with thousands of base pairs. And if you look at the dark blue bars, the dark blue bars represent a splicing ratio of 1, meaning that all the spliced reads that we detected, all the reads that we detected by Netsig were spliced. You can see that it's a short proportion of reads that are spliced, but we do see spliced reads completely, we do see these introns completely spliced after the polymerase transcribed the splice site. However, this is not the case for all the events, because we have many other cases where transcripts are partially spliced, partially unspliced, and we have many transcripts that are totally unspliced. So this is splicing ratio less 0, means that none of the Netsig reads are spliced, and this is in between. So what we see is a very heterogeneous landscape where we can have immediate splicing, fast splicing, but not all RNAs are immediately spliced. So this clearly argues against the model that long introns require exon definition. And this is very interesting because in the splicing field, this means that we have to come up with alternative models to explain how these very long introns are efficiently recognized by the spliceosome without the need of the auxiliary proteins that bind to the exon. So this is some really a result that opens up new venues of research. This is what we obtained in Drosophila, but in the meantime, in Nick Proudfoot's lab, efforts were being developed to adapt the Netsig protocol now to overcome this problem of not being able to detect spliced reads. And one of the limitations of the original Netsig protocol was that because Taka was using MNAs to solubilize the polymerase, MNAs was digesting the nascent RNA. And therefore, by digesting the nascent RNA, maybe we didn't have enough length of nascent RNA to be able to detect where the splicing had occurred. So Taka put a lot of effort on finding alternative protocols to solubilize the nascent, the complexes of polymerase with nascent transcript without using MNAs. And after many attempts, he came up with an alternative method that also starts with isolation of chromatin, but then he uses urea and a detergent called Empigen to solubilize the complexes and then to do the pull down with antibodies against the polymerase. But now the advantage is that the RNA has not been cleaved. And he uses DNAs to specifically digest the DNA in the chromatin. Now he does the immunoprecipitation with Pol2 antibody, and he just focuses on preparing libraries for the nascent RNA. And we were very pleased to collaborate in this project, thanks to a PhD student from my lab, Rui Sousa Luís. Rui is a bioinformatician, and he has been involved in the analysis of the data produced from this new approach developed by Taka, that Taka called this new approach point-seq, and point stands for Pol2 associated intact nascent transcript. So the big difference now is that the nascent transcripts are not digested by mRNAs, and therefore they are more intact. And what we did was now to analyze, to sequence these intact nascent transcripts using the Nanopore sequencing technology. You probably are familiar. This is the third generation of sequencing methods. It's a single molecule based method, and it basically reads nucleotide by nucleotide all the sequences that are present in directly on an mRNA or indirectly in a cDNA. And you can see that although Nanopore can produce very long reads up to 10,000, these are exceptions. In the vast majority of reads that we were getting with this technology was in the range of 1,000 nucleotides. But 1,000 nucleotides is way longer than the previous reads from classical Netsig, that is in the range of 60 up to 100 nucleotides. So this is now a typical, a representative image of Nanopore long read analysis from a human gene. And so the color code here is the blue reads are unspliced, the red reads are spliced, and you can see the end of the read corresponds to the position of the polymerase, and you can see polymerases were elongated, this exon in the middle of the gene, and the transcript associated with these polymerases is already spliced. In all these cases, all these red reads represent cases where splicing occurred as the polymerase was transcribing the next exon, indicating again evidence for fast splicing. So it seems that, like in yeast, like in Drosophila, mammalian cells also, and in the meantime, Carlo Neugebauer also published another study in mammalian cells, again showing evidence for fast splicing. It seems that splicing can indeed occur very fast, but this is not the whole story, because like I showed you in Drosophila, yes, splicing can be very fast, but splicing is not always occurring fast, meaning that many transcripts remain unspliced. And a very similar result was observed now using the Nanopore long reads in mammalian cells. You can see here the proportion of spliced and unspliced reads that we were detecting in this analysis. So these are transcripts. These are transcripts that the polymerase has already reached the end of the gene. So the transcript is already reaching the poly-A site, but it has not been cleaved. So the RNA has not been released from the polymerase, and it is completely unspliced. So this raises a question. This shows us, these results, together with results from other labs, are showing us that the nucleus is full of unspliced transcripts. We still don't know if these are short-lived and they become eventually processed. Maybe they take longer to be spliced, or they eventually disappear and are degraded in the nucleus. All these are open questions, but I would also like to highlight two papers that were recently published in Nature and bring up a new idea for the role of introns. Maybe introns do play a role as non-coding RNA, and they are not just to be excised by splicing and to be immediately degraded. In fact, these two papers that were done in yeast show that introns can act as non-coding RNA that may trap the spliceosome and decrease splicing as a mechanism to adapt to starvation. Yeast cells that are deprived of this mechanism, they don't survive during starvation, whether the wild type yeast do survive. So I think this is interesting to raise the possibility that unspliced transcripts in introns in RNAs in the nucleus may play some role that we are still just starting to think about. And finally, I would like to discuss whether it is important, whether does it matter to splice fast or not to splice so fast, or not to splice at all. And in this regard, so we don't know the answer to this question, but in this regard, I would like to remind you that introns, particularly very long introns, they often contain premature poly-A sites. So poly-A sites are the sequences that mark the end of genes, and they are normally recognized at the end of a messenger RNA. They are by a dedicated machinery called the 3' end processing machinery, and the RNA is cleaved, and then a poly-A tail is added. This is the normal poly-A site at the end of the message. But there are some so-called cryptic poly-A sites embedded in introns. Normally, these poly-A sites are just removed because the spliceosome will excise the intron, and the fastest this reaction occurs, the less the probability that these intronic PAS sites are recognized. And if cryptic poly-A site, an intronic poly-A site, is recognized by the 3' end machinery, what happens is that the transcript will be truncated and terminated prematurely. So this should be the normal messenger RNA that would give rise to a protein. If this intronic PAS site is used, then the messenger RNA is much shorter and gives rise to a shorter truncated protein that can have completely different function. So maybe there is a kinetic competition between removing an intron with these cryptic sites, or if the intron is not removed fast enough, these cryptic sites may be recognized and lead to premature termination. And we were very excited to see some data that suggests this may be the case, at least for some genes. So the experiment that we did here was to treat cells with a splicing inhibitor called PLA-B. It's a drug that binds to a component of the spliceosome and blocks the spliceosome assembly. So splicing does not occur. And after a short incubation of 4 hours with this spliceosome inhibitor, when we did the point-seq analysis, you can see that for many genes, now the transcript was being terminated as the polymerase was transcribing just the beginning of the transcriptional unit. So this represents the transcription start site. This is the transcription end site where the transcript should be terminated. This would be the normal transcript, but after PLA-B treatment, many genes are prematurely terminated. So as the polymerases reach this part of the transcript, the transcription stops. And what we realize is that this premature termination is more frequent in longer genes. You can see this is where no premature termination is observed. And when we see premature termination, the genes are always longer. This could be because longer genes have longer introns. Introns are the most important components to make the size of a gene. And longer introns have a higher probability of having cryptic PAS sites. So we are excited with this result because it is consistent with our idea that there may be a kinetic competition between splicing and other processes, or the co-transcriptional processes. Another question that we are very interested in understanding is whether fast splicing is somehow regulating, or it's just a stochastic process that sometimes the spliceosome is able to identify immediately that an intron is there and the intron is excised. Sometimes the spliceosome is not that efficient, and if it lets the intron be there for longer. Are there specific events that are more fast spliced than others? Could intronic polyadenylation be enhanced by inhibition of fast splicing? Can we find ways of specifically interfering with fast splicing? All of these are questions that we are currently very interested in pursuing. And related to these questions, we are also very interested in trying to understand what is the interplay between splicing, namely fast splicing, and the formation of R loops. This is a result that I find fascinating. This is an experiment, a set of experiments that were published in 2017. And what these groups did was to remove introns from genes that normally have introns in yeast and introduce introns in genes that normally are intronless. And what they found out is that deletion of endogenous genes increases R loop formation, while insertion of an intron in an intronless gene suppresses R loop. What are R loops? R loops are formed by the base pairing between the nascent transcript and one of the strands of the double-stranded DNA in the gene. So what happens is that the nascent transcript has a sequence that is complementary to one of the strands, obviously, and this sequence can invade the double helix and form base pairing with one of the strands of the DNA. This causes the other DNA strand to become single-stranded, and obviously a single-stranded DNA is highly unstable in the nucleus. And so the formation of these so-called R loops, loops caused by RNA hybridization, are very genotoxic and are known to be cause of genomic instability. And what is also interesting is that there is more and more evidence that cancer-associated mutations that interfere with components of the spliceosome, they induce accumulation of R loops, and probably this is one of the mechanisms by which mutations in these proteins of the spliceosome are driving oncogenesis, is by the genome instability caused by the formation of these R loops. So a question that we raise is whether this fast splicing, this ability to quickly remove an intron from a pre-messenger RNA could be important to prevent the formation of R loops, because if you remove introns quickly, then you stop the possibility of the nascent transcript invading the DNA, because without intron, the nascent transcript is no longer complementary to the genomic sequence. So with this, I hope I have stimulated your curiosity about the dynamics of splicing and how the dynamics of splicing may be important for genome stability. And I would like to end by acknowledging all the people in my lab that have been involved in this work. And as I mentioned, this was a very close and very fun collaboration with Taka and Nick in Oxford. And this is our funding agency. And thank you very much for your attention.
I was fascinated by this experiment where you showed that actually only a very small fraction of RNA get actually spliced. And you showed this for the ACTB genes, which is a gene which is a housekeeping gene, which is highly transcribed. So my question is, did you look at tissue-specific gene for which we know are transcribed at very low frequency? So if a gene that is transcribed at a very low frequency is spliced with a very low efficiency, this would give basically a null probability of having a spliced RNA at the end.
This is a fantastic question. Obviously, it all depends on the stability of the messenger RNA in the cytoplasm, right? Even if you produce just one correct splicing every few minutes, if the RNA is very stable in the cytoplasm, this could be enough to make the protein that the cell needs. This is a great question. We have not yet addressed tissue specificity. All these experiments were done either in drosophila embryos, where it's just housekeeping genes that are being activated. In the mammalian, the human, were done on HeLa cells with all the caveats and restrictions of HeLa cells.
But for example, for the gene encoding transcription factors, it is said that the burst of transcription gives rise basically to one RNA molecule. So, and there might be, I don't know, 10 bursts per cell cycle, so only one burst per hour. So this would really give rise to very low number of RNA. Even to the point that it's hard to believe that any single complete and spliced molecule ever reached the cytoplasm. So,
Yep. I totally agree. We need to look now to specific genes. because some genes may have some form of regulated fast splicing to ensure that they produce spliced products more often, as you are saying. I totally agree with your view. We need now to move to more tissue-specific cells and tissue-specific genes, and seeing how this differs now from gene to gene.
Maybe also related to this question, do you see, if you look at different genes, that the ratio from spliced to unspliced is differing, or is this ratio rather similar from gene to gene in the same cell?
Yeah, so first, we all of these is not single cell analysis. So because it's bulk, we cannot say that this is the same transcript in all of the cells, or some cells may have one form or another. We have tried hard to find correlations between which genes are, which genomic features are more associated with fast splicing, except for GC content, but GC content correlates with basically everything else. Except for that, we could not find yet some strong features that we would say, oh, here we have a marker for fast splicing. That's why I keep open the possibility that these may be, at least for the majority of genes, this may be stochastic. And there is a very nice paper in Cell from Dan Larson very recently, already this year, and he did real-time analysis. He also observes lots of stochasticity in the splicing decisions. I think we have to keep our mind, like transcriptional bursting, there may be also a kind of splicing burst where sometimes you splice, other times you don't splice.
I just had two very short questions. The first is, maybe I missed it, but how do you think that not splicing actually increases the risk of DNA RNA hybrids? Is it just because the RNA is longer if it's not spliced, or is it something about the sequences of introns that, you know, you have a higher chance of forming an RNA DNA hybrid there?
This is hypothesis. Our hypothesis is that it's not the size, but the sequence. Because you have these long introns, most human introns are all very long, and therefore it's the sequence of the intron that can base pair and form an R loop. If you remove the intron, then the exons are too small to hybridize and invade the double-stranded DNA.
Great, thank you. And my second question was really short. I was trying to come back to the very beginning of your talk, where you were explaining all of the difficulties that you had in the beginning with Netsig to detect spliced, transcripts. But then in the second part of your talk, you concluded that splicing is fast, right, in human cells. And so I was just wondering, is it still fast, but not fast enough to be detected by Netsig, or?
So the way we circumvented the problem that we had in the beginning was first to change from human to drosophila. And in drosophila, the introns tend to be shorter, so therefore you detect spliced reads without the need of very long reads sequencing.
But then by point-seq.
But then by point, we got rid of that limitation. So now Netsig is no longer a limiting factor because we have point-seq. And with point-seq, we have long transcripts.
I was wondering, again, on this nanopore sequencing technology, are you able to capture isoforms in this, with that, in the sense that some exons are there or not? I mean, normally you should be able to see that, no?
Yeah. So we didn't focus on that because we just wanted to measure splice or not splice. but Nanopore is widely used to detect messenger RNA isoforms. In fact, is the best method to identify isoforms, because with RNA-seq, when you have genes with the multiple exons, you cannot tell, because the Illumina sequences are relatively short. You can only detect 2 exons ligated, but you don't know if those 2 exons are ligated to exon number 3 or exon number 5. What are the other exons that is also in that particular isoform? So the long reads, either from Nanopore or PacBio, are now the gold standard to identify alternatively spliced isoforms, because you can see ideally the full length, not yet, the technology is not perfect yet, but at least you can get much more exons together, and you can be sure what is the order of the exons that have been linked to each other.
And regarding the stochasticity that this technique allows you to capture, I mean, did you try to quantify a bit more this stochasticity in terms of also in terms of frequency of having an intron which there are not, or also the fact that the stochasticity may depend on the locus or on the gene? So did you try to look closely at that?
Yep. So these are all issues that we are trying to solve. We still don't have data, but these are all the issues. These are all the questions that we are trying to figure out. Thank you for your questions.
And maybe a very last one, and I guess it's also related to the stochasticity. So you are talking just before about this idea of bursting of splicing. Mm-hmm. Do you think it has also a relation with the 3D local organization of the chromatin, and this idea of splicing factories and all these things? I mean, how does it?
I do, because but these are just my ideas. You remember I told you that human genes have these very long introns. So you need to somehow keep the exon that was transcribed first. You need to keep it close to the polymerase, so that when the second exon comes, they are just next to each other. So I imagine that the 3D structure in the nucleus is fundamental to keep the 2 exons together and allow the intron to make a loop out. Mm-hmm. that doesn't perturb, that doesn't interfere with the spliceosome. Now, what is keeping? First of all, we need to demonstrate that indeed introns make loops. We need technology to show that your field, the chromatin field, is leading on identifying technologies to look at loops. I hope the RNA field will soon be able to characterize the 3D structure of nascent transcripts in the nucleus. We are not there. We need technology to do that. But I bet my idea, my model is that introns are forming loops in the nucleus.
And in high-C, you don't see, in high-C, you may capture this type of loops, or it's too coarse to basically capture that?
It's too coarse. We tried. I mean, I would love to collaborate on something, on projects on this of this kind. So far, we have not been able to fine-tune, to have resolution of intron size. But if you have any idea in that regard, please check it. I would love to see evidence for intron loops.
Okay. Thanks, Carmo.
Thank you, Daniel.
Yes, I have a suggestion here.
Yep.
Actually, at the same time, IC was evolved. We evolved anchor dam in yeast to study very long-range interaction, and that's because for the interaction we were looking at, the interaction was much too fast to be detected with formaldehyde cross-linking. So the idea of anchor dam is that you recruit a modification enzyme that would modify the nucleic acid around, but that would modify also any nucleic acid that will come and interact with the site of recruitment. So maybe you could use this because this is not dependent on the distance or whatever, more or less. And you may imagine, I know there are enzymes that modify RNA, but you wouldn't want to use an A6 modification because it's already there. But there might be some RNA modifying enzyme that you could sort of recruit at some point and try to see whether there is a piece of RNA that come and interact through the modification.
Yep, definitely. Thanks. We need to dig it out. Yep.